Hi, everybody. Today we're going to talk about split predicates. Specifically, the FTA guidance on substantial equivalence has an actual definition for what a split predicate is. And if you aren't familiar with the FTA guidance on how they determine substantial equivalence, and you're not familiar with the term with split predicates, if you check on the link down below in the description, I've provided a download link right from our website, so it should be very fast. You should just be able to click on that and download the document. It's a let's see, how many pages is this guidance here? 16 pages. So a 16-page guidance from the FDA on substantial equivalence, and it explains to you what the FDA's process is to decide whether your device is substantially equivalent to a predicate or not substantially equivalent. And anybody that knows regulatory and knows FDA at all, they love their acronyms. So we have an acronym for substantial equivalence, SE, and we have an acronym for non-substantial equivalence, NSE. What you don't want is an NSE letter. The non-substantial equivalence letter says, sorry, we don't think that your device is equivalent to the predicate you've chosen. And a lot of people, they get very scared when they see this letter because the boilerplate language that the FDA uses says, oh, this is an automatic class 3 designation. They all of a sudden think their device has been reclassified as class 3. No, you just didn't make a good enough argument for why your device is equivalent to a predicate. So I'm going to walk you through the rules, and then we'll get into the specifics of substantial equivalence and split predicates. Because a lot of people do this, it's very routine, and I think the examples I'm going to give you are very clear, and I'll tell you what we use as a strategy. Not all our clients listen to us, but they usually end up where we recommend them. So number one, the first step is the device that you have chosen as a predicate a legally marketed device? Now, there are two ways you can look at this. Number one, is it registered and listed for the FDA? If it's in the listing for your competitor as a legally marketed device, then that's definite yes. However, sometimes a startup company will get a 510K clearance for something, and then they'll run out of money, or they're not ready to launch yet. And for whatever reason, you wanted to use that for a predicate, but they haven't listed it yet. That doesn't mean you can't use it, but the best policy is to ask the FDA during a pre-sub. You can still go ahead without a pre-sub and you can try to use that as a predicate, but you might end up redoing all your documentation claiming a different predicate, which is going to cause a delay in your submission, and you're not going to be happy with that. Plus, it's more work for you. So if you can do a pre-sub and verify that that predicate is a legally marketed one that you can use, even though it's not listed yet, the FDA will give you the answer yes or no. Sometimes we do get responses back from the FDA, no, you can't use that one. And usually the reason why is no, that was withdrawn from the market because there was a safety issue with the design that we found out about later. So that does happen. Number two part, and this is a flow chart that they take you through. There's a diagram with a flow chart, and the FDA reviewer actually will circle, this is the way we have to go, and these are the decisions we made. The second one is, what are the indications for use for this device? If the indications for use for the device are the same, so they're identical, word for word, except maybe the name of the device, then you should have no problem. This includes the RX only versus OTC. You have two little check boxes at the bottom of the indication for use form. This is FDA form 3881. If you don't check the same boxes as the predicate, or you have different wording in there for the indications, you have to justify those differences. The best policy is to go exactly word for word. Some people want to use different wording, but that doesn't help you get your 510K faster. It makes it more challenging. Sometimes it's worth doing the extra effort, but most of the time it's not. After all, do you really think that people are saying, well, I like this indication for use better than this one, so I'll buy this product over here? That doesn't actually happen. if it's not indicated for something, if it were contraindicated, somebody said, don't use it for this, that might stop somebody from using something. But you don't make decisions because you like their indication better. You use the device because it's right for what I need to do for this patient or how I'm going to treat somebody. The third question in the substantial equivalence flowchart is the device that you're using have the same technological characteristics? If the answer is yes, that makes it easy. You don't have to go any further. You stop at step 3, 510K substantial equivalence. So Wandy, one of our new regulatory consultants, he recently did a submission for some bipolar forceps, and they were literally identical. We had a client that knew exactly what all the different competitors were. He had used them in surgery, and he's like, I know this one is exactly the same as what we've made. It's just from a different contract manufacturer. So that made it really easy to say it has the exact same indication for use, the exact same technological characteristics, and he even was able to avoid a lot of testing that you would normally have to do because it was identical. Same materials, same length, same shape, same thickness. Everything was the same. It was so easy. And Wandy and the client got rewarded. 90 days later, boom, you got your 510K clearance. And it didn't go beyond the 90-day mark. And you didn't have delays where they had requests for additional information. It was an interactive review. It was fantastic. But that doesn't always happen. Most of the time, you actually have the answer is no, it's not the same technological characteristics. And that's when you have to go to question 4 and things can fall apart. But that does not mean as a company, you should make your decision on substantial equivalence based on question 3. You don't want to go out there and just try to find something with the same technological characteristics because that's not the most important thing. The most important thing is that it has the same indication for use. So if you have different indication for use, don't use that predicate. And this is how the split predicate arrives. A company, and I'm going to use my as an example here. I know not everybody does IVD submissions, but I think it's a really simple example that everybody should be able to understand. When everybody on the planet got COVID, and just about everybody on the planet did get COVID, you had 2 different types of tests that they could do for COVID. One was a molecular test. The molecular test looked for the DNA of the COVID virus. The other test was an immunoassay or an assay looking for, do you have antibodies to this? So you're going to actually take a sample and you're going to put it on this little IVD test, and you're going to see if you have 1 line or 2 lines or 3 lines, because they would have one for the control, and then would they have a second control, or sorry, one for the control, and then they would have one factor that they were looking for and a second factor. So by having both factors in there, they increased the effectiveness of that test. But this is looking for little lines on a strip versus looking for DNA. So an immunoassay is not the same as a nucleic acid test or a molecular test, and the FDA preferred to have companies get cleared and to preferentially get these molecular tests out there because they're more accurate. So completely different technology is used in these 2 different types of IVD tests, but they were both looking for, yes, you have COVID, or no, you don't have COVID. So the indication would be identical for these 2 products, but the technology is totally different. So one company is looking for what little pieces of DNA are you looking for, and are those pieces of DNA unique to this COVID-19 strain, or is it ubiquitous and it's in all COVID strains? Because COVID-19 wasn't the first time COVID came out, it was just that COVID-19 strain was a problem. And then they had a whole bunch of variations that popped up over time. That's how viruses work. So they were very concerned about, what little pieces of DNA, what sections of DNA are you looking for? What genes are you looking for? Whereas in the immunoassay, they were looking for something completely different. And so that's the difference in technology. And you can't say, well, I know what I'm going to do. There's no 510K clearance or de novo approval yet for an immunoassay, but they have exactly the indication for use I want. So I'm going to use the indication for use for BioFire's um, test, which is a molecular test. And I'm going to use the flow through assay over here, this immunoassay for the technological characteristics. So I'm going to list two predicates. That's all I got to do, and this will be substantial equivalent. And that won't work. That's a split predicate. You've taken the indication for use from one device and taken the technological characteristics from the other device, put the two together, and said, this is my substantial equivalence argument. Two predicates instead of one. That's not allowed. That's called a split predicate. You got to have the same technological characteristics and indication in the same device. So when you would use multiple predicates is when you have multiple indications. Or here's another possibility, you have more than one technological characteristics with the same indication. So you're going to do it more than one way. Most of the time, when we have IVD products go on the market, the reason why a brand new IVD test has to go the de novo route, like BioFire had to for the COVID-19 molecular test, it's because even though BioFire had tons of molecular tests out on the market that they were going to bundle together as a kit or a system, they didn't have one for that specific indication, the COVID-19 virus. So since they didn't have that indication, they had to go down the de novo route. They failed at question 2. They didn't have any, even though they had lots of devices out there that they made themselves that were on the market legally, none of them had the same indication, even though they had all the same technological characteristics. So that's why they weren't able to go the 510K route, and they had to go the de novo route. But you can't do that. You can't combine. We've got this 510K technological characteristics over here, and this one for the indication. I'll put the two together. I have two predicates, and we're good. Historically, a lot of companies would list multiple predicates with the theory that if I can show how many products are like mine, I'm more likely to get my 510K clearance. That doesn't work. Companies did that, but it doesn't help you. It makes it harder. It actually, it's unlikely that 3 different devices are all exactly the same as your device. This part of my device is like this brand, this part of my device is like this brand, and this part of my device is like that brand. You're making this harder than you need to make it. Pick one predicate, just one. And if you have time for a pre-sub, and you really should make time for a pre-sub, ask the FDA if there are any concerns of differences in the technological characteristics that you might have outlined. Here are the differences. Here are our justification for why we don't need to pick a different predicate. We can just do verification and validation testing, showing that those additional technological characteristics don't create any problems. Because of that, we don't have to have another predicate. We just provide the verification and validation testing. And that's covered in question 5 of the substantial equivalence comparison flowchart. So that's the right way to go. The right way to go is pick one predicate and justify the technological characteristics. Don't cause new risks or present any greater risks for safety and efficacy because you've done some additional verification testing. A simple example would be if I add Wi-Fi to something. The predicate didn't have Wi-Fi. I'm adding Wi-Fi. It's a technological characteristics. I do not need a second device as a predicate that does have Wi-Fi. I definitely don't want to pick one that does a completely different indication for use. Then now we're talking split predicates. All I have to do is say, look, there are many other devices on the market that have Wi-Fi, and this is the testing that they have to provide to the FDA to show that they're safe and effective. So we're just going to do the Wi-Fi testing to cover the Wi-Fi functionality, but otherwise it's a standard 510K submission. So in those cases, the FDA may say, oh, please provide a reference device that has Wi-Fi. Sometimes they ask you to do that, sometimes they don't. But don't offer up. And we've got all these reference devices to cover all the new features we've added. You're making it too complex. One predicate in verification testing to cover technological characteristics that are different. And there's probably a standard that the FDA recognizes, and there's probably a guidance document that the FDA recognizes, like EMC testing, wireless coexistence testing. There are tests out there to address the Wi-Fi. You don't have to pick another reference device or another predicate. Let the FDA tell you you have to do that, or ask the question: is it sufficient to use this approach, or do we need to add a reference device? Let them answer that question, yes or no, and explain why. Don't offer up, let's make this as complicated as we can, and raise a whole bunch of questions you didn't need. So I hope I explained well enough for everybody what the difference is between substantial equivalent. It has this. It's a legally marketed device. The predicate is. It has this. Your device has the same indication for use as the predicate, and third, it has the same technological characteristics. But if you don't have the same technological characteristics, don't try to add on more predicates, predicate number 2, 3, and 4 to fix the problem. Identify the tests that you need to use and run those by the FDA to address these differences in technological characteristics. This is how other products have done testing to show that it's safe and effective. So we're proposing to do those tests. Will this be okay? And they'll say yes or no. And they'll let you know whether you need a reference device. And in the worst case scenarios, they could say, oh, you need a second predicate. And because I have a little bit more time here, now I'm going to cover one more thing for you. When do you need more than one predicate? The time that you need more than one predicate is when you have more than one indication. A perfect example of this is multi-parameter monitors. Another example in the IVD world would be when you're looking for more than one microbe or more than one virus or more than one analyte if it's a chemistry test. When you're doing more than one thing, more than one indication, that's when you need more than one predicate. And you have to identify what the primary predicate is. And whatever that 3-letter product code is, that's the primary predicate, 3-letter product code. And you're going to put those 3 letters first in your string of classification codes. And that's the group at the FDA that's responsible for your submission, the review panel. And some of those other codes might be in other divisions, and they can always ask somebody, or they might be able to review it themselves, but you have to have a primary. And you want to use as few predicates as possible, even though it has multiple functions. Don't try to find one predicate that has almost everything you want, and then just add one more if you have to. So that's a strategy we see a lot in multi-parameter monitors. Multi-parameter monitors could have 4 or 5 different functions. So a multi-parameter monitor would be something you see at the bedside, like a Welsh Allen unit or a Philips unit, and it would measure your temperature and your blood pressure, and it would measure your ECG, so your heart rhythms. It would measure pulse oximetry, so your SpO2. And you might even have a really fancy one here that's for home use or in a nursing home that you can also tie it to your scale, so it'll measure your weight and track that. All these other things. It could have blood glucose. You can add tons of things to these multi-parameter monitors. They want to have like a hospital lab in a cell phone. This is my new multi-parameter monitor system. They can do that, but think of all the different indications that has now. Every single one of those you need a predicate for. So if you can find a device out there that has most of them, use that as your primary predicate, and then add a secondary predicate only for the features that didn't have. And the example that I gave you with the blood glucose, that would be pretty common. You can probably find something that has everything but the blood glucose, and then you just add on one more predicate, a secondary predicate for the blood glucose. And it's probably a module that you're adding to the design that you've licensed from another company, and you can use their predicate and all their testing data if you do it right. So whenever you're trying to do these complex devices where you've tried to put all into one and had added all these features, try to find another device that's done something similar. Have that as your primary predicate. And if there's something left out, add on a secondary predicate if you have to. But that should be the topic of discussion in a regulatory strategy meeting with the FDA, i.e., a pre-sub meeting. So I hope that helps you understand when you would use more than one predicate and what a split predicate is. The split predicate is you have one device is the same for indications and another device is the same for technological characteristics, but neither of the two devices you're proposing has both. And to be substantial equivalence to a predicate, it has to have both. Okay. I think that answers all the questions. As I said, there's a link down below to the FDA's guidance. I'm hoping the example I gave you is a little more clear. Sometimes people have trouble with the FDA examples that are crystal clear to the FDA, but not so much to anybody else. But I hope that helps. And if you still have questions, don't hesitate to put comments down below. Every Friday after I do this, I go into the comments and I try to answer what was put in there in the last week. And you can always go to our contact us page and schedule a meeting with me if you need some help. And identifying predicates and regulatory strategy is what our company does. So we'd be happy to help you with that. And a lot of times we can do the very simple stuff right on the call with you during your free introductory call, and give you an idea of how we work and how you do these the predicate selections and how you identify the regulatory pathway. I hope you have a great weekend and see you later. Bye bye.